On behalf of the Association of Migraine Disorders, I congratulate all the applicants and finalists of the second annual Migraine Science Collaborative, or MSC, Neuroscience Poster Competition. This would not be an event without the efforts of all the participants. So we should ask the question, why do we have a competition focused on migraine disease? As you may know, the migraine science community is vibrant, but very small. This is a head scratcher because migraine disease and other related forms of chronic but intermittent pain are widespread and disabling conditions. They may not kill you, but migraine symptoms, whether it be a headache, dizziness, extreme fatigue, or something else, suck the pleasure out of living sometimes. Also, these are health conditions that have enormous cost to business productivity, demands on our healthcare system, misuse of limited resources such as antibiotics. AMD is fortunate to work closely with many clinical and basic researchers from the migraine community. And as partners, we're determined to raise awareness in the much larger neuroscience community and let them know that migraine can be a fascinating and rewarding career path. With this annual competition, we strive to highlight some of the best research projects by the brightest emerging scientists that are either directly advancing our knowledge of migraine disease or indirectly, because while some of the research projects may not have been designed with migraine in mind, they all address issues that have a bearing on one or more aspects of this complex biology of migraine disease. So with that said, let the competition begin.
Our first speaker is Micah Johnson. I hope I'm pronouncing your first name correctly. Micah, please correct me if I hadn't.
Yes, it's me. I pronounce my name like Micah.
Micah, okay, I'm sorry. Welcome.
That's okay.
Okay, thanks for kicking things off.
Sure, yeah, let me go ahead and get started then. My name is Micah Johnson. I am a fourth-year PhD student in the Holtman Lab at the University of Iowa. So today I'm going to talk a little bit about my work on migraine, early life stress, and dysfunctional brain circuitry. So to start out, we're facing a problem in the migraine field when it comes to medication. We have some great treatment options, but not all of them work for everyone. So a major focus of our lab is using a brain-wide network-based approach where we focus on dysfunctional brain circuits to identify potential new avenues for therapeutics. So we know migraine involves complex brain circuitry, resulting in a variety of symptoms that we're just now starting to understand. We know that these symptoms present differently across individuals, and some pharmacological treatments work better than others. We also know from imaging studies that multiple brain regions are engaged during migraine attacks. And so our lab is broadly trying to understand how we can look at the coordinated activity of multiple brain regions at the same time to identify circuits that are regulated properly to develop new therapeutic targets that we can repair and improve outcomes in migraine patients. So how are we doing this? First, we induce a migraine-like headache, or we think is a migraine-like headache in rodents using calcitonin gene-related peptide, or CGRP, which is a peptide that's heavily implicated in migraine. So many of you are probably familiar with CGRP. We build our own electrodes with individual wires that can target up to 15 brain regions at the same time. And then we implant these electrodes into rodents to measure their electrical brain activity. And the electrical brain activity that we're measuring is referred to as local field potentials, or LFPs, that provide important signals which tell us what's happening in the brain, similar to EEG in humans. We can then break these raw LFP signals into different components that give us meaningful information. And briefly, we can look at the amplitude of the signal, which correlates with the amount of activity coming from a single brain region. We can look at coherence, which represents how well LFP signals from two different brain regions are synchronized, or the crosstalk between two brain areas. And then after we find the area of greatest coherence, we can then introduce a time shift to this area, which gives us the directionality and therefore tells us the direction of information flow across two brain regions based on which region is clearly leading that of the other. And moving on to my data, so I'm only showing a portion of what I showed at SFN and what we've collected for the sake of time, but we implanted way more brain regions than this, if anyone's interested. So interestingly, we found that the amygdala leads the thalamus, which is shown here by this peak in magnitude within a low frequency band. So it's about 2 to 7 hertz. And thus we hypothesize that this is where we might expect changes in other LFP measures to occur. And so when we looked at one area of the thalamus, the posterior thalamus, we found increased power. So that's indicated by this darker red signal. After we injected CGRP into our mice, which indicates increased brain activity during while the mice are experiencing what we call a migraine-like attack. But I wanted to figure out a way in which I can observe brain changes a little bit more endogenously. So I use early life stress, which Gretchen Tiagen and colleagues have repeatedly demonstrated that experience of early life stress or early adverse experiences predisposes humans to migraine in adulthood. So to mimic that relationship in mice, I use maternal separation, where I separated pups from their mothers for 3 hours each day, and I limited their enrichment for the first few weeks of life. Then I tested their migraine-like behavior in adulthood. I don't have time to explain all of these behavioral tests, but to briefly summarize the results, I saw an increase in anxiety-like behavior, photophobia, which is light sensitivity, and then also mechanical allodynia, which is pain to touch that isn't normally painful, in my animals that underwent early life stress and now were treated with CGRP. So my big picture goal here is to be able to do this early life stress paradigm in my animals and record their brain activity, compare it to what I'm already seeing in animals that we're inducing the migraines in, and then determine what brain connections might explain this increased susceptibility to migraine in adulthood after experiencing early life stress. This is particularly important because early life stress is something that most people don't really have much control over. So I ask, what if we can identify where the brain circuitry is dysregulated in this population, repair these dysfunctional brain circuits, and therefore improve outcomes for migraine patients despite experiencing stress that's out of their control. And so to conclude, let's take the stress out of migraine treatment and repair dysfunctional brain circuitry to improve the outcomes of migraine patients. Thank you for your time.
All right, great. Thank you so much. That was fantastic. A great way to kick things off. So our next speaker, and again, for all the speakers, if I mispronounce your name, please correct me at the beginning, is Aldalil Lorian from INSERM. Excuse me, Lorian Delay, I'm sorry.
Thank you so much, and thank you for to be here tonight. So my name is Lorian Delay. I'm a postdoc in Physics for Medicine lab in Paris. In this lab, they develop a new approach of imaging, that is the functional ultrasound imaging, developed by Michael Johnson. And I use this technique to understand functional brain and trigeminal changes in migraine. So very quickly to answer to your first question, so migraine is a complex neurovascular disorder with a complex pathophysiology that we still need to understand. So there is more women affected than men, and it's characterized by headache, crisis, and sensory sensitivities such as photophobia and allodynia. So the objectives of my project is using relevant rats models of migraine that we want to compare in both male and female to assess a potential sexual dimorphism. We aim to define the vascular dynamic changes, the functional connectivity in a large part of the brain. And we ask also the question, how anti-migraine therapies can impact this dynamic. So here today, I can just explain like very quickly one of the migraine model that I'm using that is induced by a repeated injection of nitric oxide donor, the isosorbate dinitrate, in rats. And first of all, we were able to separate two subpopulations, as is relevant also in clinic. So as you can see here, when we are calculating the mechanical sensitivity using the bone Freiherr test that we are applying on the periorbital area, we were able, in comparison to saline, to see that there is a sensitive rats to this molecule. That's around like 80% of the rats, and there is a decrease of the mechanical threshold for these rats. And there is also like ISDN non-sensitive rats in around like 20% of the cases. And this is for it's really relevant to have two populations. The technique that I'm using is based on plane waves instead of being focused on point in comparison to conventional ultrasound. And the power of the player that's proportional to the number of red blood cells in a unit volume. It's increasing a lot the spatial resolution and temporal resolution in comparison, for example, with fMRI. So here, what I wanted to show you, and I think it's really, really interesting, is in comparing sensitive rats, so that are like painful, and non-sensitive rats, we can really see that in baseline, I don't know if you can see my in baseline, we have not a lot of change in cerebral blood flow, but in sensitive rats, when we are injected the 5th injection of ISDN, we can really see that my animals, sorry for that, I don't know. We can really see here and here that there is a big increase in cerebral blood flow just following the injection that is not seen in non-sensitive rats, even if we are injected ISDN. And the most interesting thing is that we are able to induce repeated crises of migraine headache, as we can see here. And the big increase of CBV are seen in the pain matrix area of the brain and the area of the brain that are also correlated to photophobia. So here is the example of the injection for non-sensitive rats. So what I wanted to point today is that I really think that the functional ultrasound imaging will add really new insights in migraine studies, because it's portable, easy to use, cost efficient, and it's a very high spatiotemporal sensitivity. In preclinic, the big advantages is that we can use this technique in awake and freely moving small animals, and there is directly a clinical perspective. As you can see, I don't know to. So this technique is validated in human. We can assess effectively cerebral blood flow changes that are associated directly via the neurovascular coupling to the brain, to the neuron activity, the functional connectivity. We can also assess what we know as a response to functional stimulation. Here is an example of the specific activation of the trigeminal ganglia using von Freiherr filaments. And also here is the activation of the superior colliculus after visual stimulation. So we are pretty sure that we will add new insight in migraine, and to hope we will use this technique also in the clinic. And there are also like a perspective for neurostimulation, but it's too short to explain everything that we can do with this technique. So I really want to thank you for your attention.
Great. Thanks so much, Lariane, for that great presentation. So it looks like next up we have Victor Chua here, I believe from University of Utah.
Hi, my name is Victor. I'm a graduate student at the University of Utah, and I recently started working on this project on migraine, focusing on the neurons in the trigeminal ganglion and its role in migraine pathophysiology. So just a brief background, why the trigeminal nerve. So the trigeminal nerve innervates the craniofacial region. It relays sensory information from this region to the brain, and that makes them a key element in pain sensation related or associated with headaches such as migraine. This trigeminal nerve arises from the trigeminal ganglion, which houses the neuronal cell bodies of this system. And in order to identify molecular targets and develop effective drug leads for migraine, it is important that we characterize the neuronal cell types in the trigeminal ganglion and identify which ones are involved in migraine pathophysiology. So in our lab, we developed this technique called constellation pharmacology. It is a calcium imaging-based platform where we dissect the trigeminal ganglion from mice, dissociate the neurons and plate them, and classify these neurons into cell types based on 3 factors. The first one is the size of the cell. The second factor is the neuronal markers, such as the expression of CGRP and the binding to isolectin B4. And the third is the functional expression of the TRP channels, which are the principal determinants of pain, molecular determinants of pain sensation. So just how to basically read the traces. So an individual trace represent the calcium profile of an individual neuron, and a peak represents indicates the expression of the corresponding receptor of the applied agonist. By applying constellation pharmacology, we were able to identify the constellation of ion channels and receptors that are expressed by each cell type. And so far, we have identified 19 different cell types in the trigeminal ganglion. Now, the power of this approach is that it enables us to monitor the changes in these cell types when certain conditions are altered. So, for example, this is also the pilot study that I did. I wanted to look at what the effect of CGRP would be to these neurons. So I incubated trigeminal neurons with CGRP, and what I found is that CGRP promotes the expression of a ATP receptor or purinergic receptor. This is exciting for 3 reasons. First is that ATP is known as a painful stimulus. Its receptor, purinergic receptors, are known pain transducers. So the upregulated expression of these these receptors could be contributing to the pain sensation experienced by migraine patients during headache attacks. Second is that this upregulated purinergic receptor shows a distinct phenotype. So it's behaving differently from the innately expressed purinergic receptors by the trigeminal neurons. And this tells us that this receptor has a different identity from this receptor, and it could be an important receptor that that plays an important role in migraine pathophysiology. And last is that CGRP only affects select cell types. So in this graph, the orange bar represent the neurons that are affected by CGRP, and we can see that these neurons are mostly localized in or classified in these 2 cell types. And this is exciting because this tells us that, or gives us an idea that these 2 cell types are the ones that are involved in the CGRP pathway in migraine. And this would help us move forward as we can focus on these 2 cell types, characterizing these 2 cell types, as well as this purinergic receptor upregulated by CGRP for future experiments and when we eventually move to animal migraine models. So also just a final note, I just want to share another good thing about constellation pharmacology, and that's after the experiment, the cells remain viable in constellation pharmacology, and we can use these same culture with other techniques such as patch clamp electrophysiology and single cell transcriptome analysis for further characterization of these cell types. I know that was short, but I hope that I've convinced you that constellation pharmacology is a powerful tool for classifying neuronal cells into different cell types and how this approach can be applied to monitor changes during disease progression. Thank you.
Great, thanks very much, Victor. So next we have Rebecca Hornon, who is from Texas A&M University. Rebecca, please go ahead.
Hi, I apologize. I don't have any slides like everybody else. I just recovered from being sick and moving to Utah. So my research is a little bit different. While I don't study migraine, I do study trigeminal or facial pain. So in our lab, because I'm in Dr. Phil Kramer's lab at the School of Dentistry in Dallas, we study post-traumatic neuralgia by injecting human herpes zoster, the varicella zoster virus into the rat whisker pad. And in our rat model, there's also sex differences. So we focused on looking at why the females, when they're in proestrus, which is when they have high plasma estrogen levels, experience lesser motivational pain, or the diestrous females, when they have low estrogen, have greater motivational pain than proestrus females, as well as male rats. So in our study, we also looked at two different brain regions. We looked at the central amygdala as well as the parabrachial, because the central amygdala has GABAergic neurons in it that project to and inhibit the parabrachial nucleus. And both of those brain regions do modulate orofacial pain. So in a previous study, we noticed that with the estrous cycle in females, that there are some genes that are modulated by the estrous cycle. For instance, in this study, we looked at the Neurexin-3 alpha protein, that is a synaptic cell adhesion molecule. And in the proestrus females, they had a higher expression level than diestrous females. So we wanted to determine if we actually reduce the expression of this protein in the females within the amygdala, as what the proestrus females, as well as the males, if they would exhibit orofacial motivational pain similar to diestrous rats. In addition, we also looked at the GABA release to the parabrachial, because when you knock down this cell adhesion molecule, you reduce GABA expression or GABA release. So in order to target this protein, we infused a virus that reduced the amount of protein expressed within the amygdala. And then to investigate if reducing the expression of this Neurexin-3 alpha protein also reduced the amount of GABA released, we infused the parabrachial with a virus that senses GABA. So when GABA binds to it, it fluoresces. And then to see this fluorescent signal, we implanted a fiber optic lens to the same region that the GABA sensing virus is in, so it's in the parabrachium. So we found that when we infused the amygdala with the Neurexin-3 alpha short hair RNA, it did reduce expression of this protein in the amygdala. And we also saw that both males and proestrus females had an increase in the orofacial pain response, and that there was also a reduction in the GABA release to the parabrachial. So what our data suggests is that this Neurexin-3 alpha, the cell adhesion molecule within the amygdala, does modulate orofacial pain by regulating GABA release to the parabrachial, that then regulates neuronal activity of the ascending pain pathway. So pain signals that go from the site of pain to the brain. So I said that I don't study migraine. However, there are several chronic orofacial pain disorders, such as migraine, temporal mandibular joint disorder, and trigeminal neuralgia that are more prevalent in women. So migraine and TMD pain have been reported to fluctuate with the menstrual cycle, suggesting that the ovarian hormones do modulate pain. And then ovarian hormones are also known to alter gene expression, which may contribute to the fluctuations in pain throughout the menstrual cycle. So in our study, we show that altering expression in orexin-3 alpha protein, which is also modulated by the ester cycle within the amygdala, modulates both GABA activity in the parabrachial as well as the animal's response to orofacial pain, suggesting that this orexin-3 alpha protein expression is modulated by the rat ester cycle and can also contribute to fluctuations in migraine pain. So if it does actually fluctuate with by hormones, such as through the menstrual cycle, this is one protein that could potentially be targeted to help modulate migraine pain. That is all I have.
Okay, great. Thank you, Rebecca. Everyone's doing a great time keeping on time, so that is fantastic. So our next speaker will be Juliet Mirigi from UT Dallas. Juliet, please go ahead.
I'm Juliet Mirigi. I'm a third-year PhD student in Dr. Ted Price's lab here at the University of Texas at Dallas. And the main focus of our lab and my PhD research is on neuropathic pain, and in particular, the role of a cytokine molecule called oncostatin M in the human dorsal root ganglia. So just to give a brief background into why we are interested in neuropathic pain research. Neuropathic pain research engenders multiple entities and affects at least 20 million Americans. A big proportion of this population that is affected by neuropathic pain is caused due to diabetic neuropathy. And while there are a lot of currently prescribed drugs that are out there, they are either not as effective at treating pain or could also worsen the outcomes in the long term. And there is also a significance in the prescription of opioids that can lead to dependence over time. So sufficing to say, this does impose such a big economic burden. And one major interest in our lab is to develop non-opioid targets for pain that are very translatable from preclinical to clinical models of research. So to do this, we do have a collaboration with our local transplant a site where we are able to source dorsal root ganglia as well as other sensory tissue such as the spinal cord, the vagus nerve, and nodose ganglia, spinal meninges, and sciatic nerve. And we have been able to use this tissue to perform a variety of assays that have really showcased some similarities as well as differences between species, which could in turn affect the translatability of potentially great pain targets that we discover in the preclinical side. So some of the work that we've been able to do is high throughput sequencing, RNA transcriptomic sequencing, proteomic assays, as well as we are able to store or harness this tissue in artificial cerebrospinal fluid and bring it to the lab and culture it and perform live assays on it. Another source of our human tissue has been an ongoing collaboration with clinicians from the MD Anderson here in Texas. And for this, we have been able to work with a subset of clinical patients that have that undergo vaborectomy surgery to remove the dorsal root ganglia and surrounding nervous tissue, which is compressed due to a tumor growth. So we found that using this tissue and performing transcriptomic sequencing, we found a very interesting upregulation of oncostatin M, which actually sparked our research and my research into the role of oncostatin M. So we found that oncostatin M, which is a cytokine belonging to the IL-6 family, is highly upregulated with more presence or expression in male versus female patients. We also found that when we removed this tissue and performed electrophysiological studies with just the DRGs from the pain side from these patients, we find an aberrant activation or firing of these neurons, which indicate to us that this might be a driver for chronic pain in this cohort of patients. So leading up to our discovery on oncostatin M, we were interested in seeing whether the receptor for oncostatin M is indeed expressed in the dorsal root ganglia. And for this, we turned to fresh frozen tissue sourced from the STA local organ transplant site. And we were able to perform in situ hybridization where we detected OSM receptor, OSMR, in both peptidagic and non-peptidagic populations. We also, even with an N of 3, 2 males and 1 female, we were able to see much slightly higher expression of OSMR mRNA in the male tissue samples. We also performed a very high throughput aptamer-based proteomic assay where we looked at the expression of OSM protein, OSMR, and LIFR, which is also another receptor that OSM can utilize. And we were able to find a higher expression. We were able to note a higher expression of OSM in the male versus female at baseline tissue from healthy donors. So to better understand the physiological role of OSM in the periphery, we took this tissue, we cultured it, and performed some electrophysiological recordings. And here we see that exogenous application of OSM does induce firing or increase firing in the DRG neurons, cultured DRG neurons. We also took explant slices or acutely sliced preparations and treated OSM. And as you can see, there is an increase in the phospho-EIF4E signaling, which indicates to us that there's an activation of not only the neurons, but also the surrounding glia-like cells. So one of the assays that we've been able to perform in our lab with the human tissue is to look at the open chromatin accessibility and with a focus in the sex differences, because this is one of the factors that determine how chronic pain is driven in males versus females. And I think a lot of us can widely accept that there are many sex differences, including some that we have started to see with regards to OSM and OSMR. And so we performed an assay for transposes dependent accessibility of chromatin. And here we can glean that with in male samples, there was found to be more open chromatin access sites near the OSMR transcription start sites. Interestingly, some of the transcription factors that we found docked near this site were expressed selectively at particular neuron subtypes that were also correlated with the expression of OSMR, as you can see here. So we see a higher expression of OSMR and associated transcription factors in the silent nociceptors that are enriched for each markers, as well as the low threshold mechanoreceptors with higher expression in males. So even though my research does not directly tie in with migraine research, I think we can appreciate that this the applicability of this research approach to studying migraine. First of all, is that we have started to really appreciate using human tissue to drive our preclinical research. And I think it's something that should be encouraged within the migraine community. Also with an emphasis in looking at sex differences with regards to different pain targets that are identified. Lastly, one good takeaway from this is that we can use a plethora of multi-omic approaches, from transcriptomic sequencing to proteomic, as well as genome-wide studies to effectively pinpoint targets that can, that are effective and more likely to succeed in migraine trials. So with that, I would like to thank you.
Great. Thanks very much, Juliet, for that great presentation. Just a reminder, if anyone has questions for any of the speakers, please just type them into the chat box, and you could just indicate which speaker you may have a question for, and we'll take those questions at the end of this category, which is physiology. So I'll introduce our next speaker now, Ariaj Bizanti from the University of Central Florida. Please go ahead.
Thank you. So I'm Ariaj Bizanti, a PhD candidate at University of Central Florida in Dr. Cheng lab. I'll be talking how chronic intermittent hypoxia induced remodeling of the autonomic innervation in the mouse heart and how this is associated to migraine and pain. So first, let's talk about that association. So intermittent hypoxia is a hallmark of obstructive sleep apnea, a very common disorder that affects people's breathing while they are asleep. A lot of people are unaware of it. It's been shown that intermittent hypoxia induce migraine-like attacks with and without aura and dilated cranial arteries. And when it triggers that migraine attack, that's independent of any pharmacological agents. In the arterial desaturation in patients with intermittent hypoxia has been associated with increased pain, and that was independent of the sleep fragmentation or inflammation. And lastly, both intermittent hypoxia and sleep fragmentation has shown to be enhance the pain and may potentiate opioid analgesic effects. So what do we do in our lab? So our goal is to map the innervation of the heart and how it's remodeled in different diseases. Specifically here is the chronic intermittent hypoxia. So first we take the C57 black mice, we harvest the heart, we separate the atria and the ventricles and cautiously remove the attached organs, but without any sectioning to avoid any disruption of the continuity of the neural network. So we take that intact tissue, we stain with different markers, including tyrosine hydroxylase, TH, that's a sympathetic marker, calcitonin gene-related peptides, CGRP, a nociceptive marker, and VAC, a vesicular acetylcholine transporter, a parasympathetic marker. And then we image the tissue. So we stitch it together, just the right atrium. We create 700 images and put them together to create that complete map of the neural network. But at the same time, we keep it at a cell axon varicosity scale. So if we zoom in, we can still see the detailed innervation. And we expose the mice to either room-air or hypoxia with a profile that mimics the obstructive sleep apnea. And then we assess the remodeling. And for the ventricles, it's few hundred microns, so it's very thick. It's been challenging, but we optimize our methods to map the whole thing again without inducing any cuts or disruption of the nerve. And then we trace the nerve with a software called Durlusa 360. And with our collaborators in Auckland Bioengineering School, we embed those reconstructed nerves into a 3D heart scaffold to create that brain-heart atlas. So why the heart? The heart, it pumps blood throughout the body. So it provides both oxygen and nutrients to all parts, to all organs, including the brain. So when there's a decreased blood flow in the brain, so there's low oxygen, hypoxia, which increase many biomolecules, including adenosine, CGRP, nitric oxide, hypoxia factor, which all disrupt the blood-brain barrier and initiate oxidative stress. And that oxidative stress create that pro-inflammatory state. So that loop between the cardiovascular and the brain has been studied for so many years. Like migraine is often seen in people who have that foramen ovale open, that hold between the left and right atria, postural tachycardia. But it's worth noting that cardiovascular diseases usually happen in the older age group. So it's more like a risk factor of migraine. But both migraine and hypoxia happen in the younger group. Both are chronic, intermittent, and are strongly associated with cardiovascular diseases. So in our research, this is a poster presented in the SFN. So we've seen that chronic intermittent hypoxia actually induces remodeling of the catecholaminergic axon innervation in the mouse atria. So this is using the tyrosine hydroxylase, the sympathetic marker. We've seen that the structural remodeling include increase in density of the innervation in many of the regions of interest. Specifically here, we trace the nerves near the sinoatrial node, which is the pacemaker of the heart. So there's an increase in complexity and change pattern, like more disorganized pattern in the around the sinoatrial node. There's also a change in the pattern throughout the 3D Z-stack model of the whole atrial muscle. Seen like the innervation of the blood vessels here, the inner lumen diameter is decreased, so more vascular resistance, higher blood pressure. We have also seen increased tortuosity, so that twisting and bending in both epicardium and myocardium. So all of this basically create that disorganized pattern and overactivity of the sympathetic system. Lastly, we have been able to map the CGRP axon innervation in the flat mount of both rat and mice hearts, and provide that complete topographical organization. We think the implication of this, by determining how chronic intermittent hypoxia remodels the autonomic and nociceptive innervation of the heart, can give us insight like how migraine can affect the neural network remodeling and improve our understanding of how the cardiac innervation relates to that migraine-induced cardiovascular diseases. All of this can potentially guide us to the development of more therapeutic targets that can reverse that remodeling. Like for example, now, like Julie talked about, the dorsal root ganglia and the stellate ganglia, which are the source of those innervations, they're already used as a therapeutic target. So their blockage are already using to treat several diseases. So by knowing and revealing how migraine remodel that innervation, we can shed light into so many unanswered questions and improve and advance our interventions to help the people who struggle with migraine. Thank you.
Okay, great. Thank you. There's actually a question for you
Yes.
from Marianne. Do you know if naked mole rats that are resistant to hypoxia and are pain-free show changes in catecholaminergic axon innervation in atria?
Naked mole rats. Never had experience of that.
Okay, it's not a problem. It was just my curiosity, because I know that it's a very useful tool for pain analysis, and I thought that they are like I knew that they are like resistant to hypoxia, very resistant. So I thought it could be like very interesting to also like ask this question in other model, like just to know, just to know. But thank you.
Yeah, it's a very interesting question. Like we actually we have a whole project dedicated to pain, so it would be very interesting. Like we inject a tracer into the dorsal root ganglia actually, and we map the CGRP differently to see how specifically that's labeled. So it would be very interesting to do that.
Thank you.
Thank you.
Okay, great, thank you. So our other speaker for this section is Selim Aldalil from the University of Florida. Please go ahead.
All right, hi everybody, just let me share my screen. Can you all see it now? Thumbs up, great, perfect. All right, just a correction, I go to Sewanee, which is an hour south of Nashville, but I did this as a part of a summer program at University of Florida. Today I'll be talking about the project that I presented at SFN, which is an investigation of the impact of chimera NDUs repetitive TBI on neuroinflammation and plasticity. So let's unpack this title a little bit, starting with TBI. TBI, or traumatic brain injury, results from sort of exposing the skull to external forces. You can think of it as a car accident or an injury in sports, for example. TBI accounts for about 2.5 million emergency department visits every year. Also, about 5 million Americans live with TBI-related disability. If we look at this figure on the right-hand side here, we can see that the two age groups that are most prone or susceptible to TBI are 15 to 24-year-olds, as well as individuals above 65-year-olds. Which could be possibly because of either lifestyle or dementia and cognitive and motor decline. So one sort of, actually the most reported symptom of TBI is post-traumatic headache as well as cephalic pain. And the difference is that TBI patients actually suffer from acute post-traumatic headache, which is, like I said, the most common reported symptom. This sort of symptomology persists for 3 months following the injury, and sometimes it can even go beyond this 3-month sort of period, resembling clinical features of migraine. So it is sort of a very common symptom in TBI patients. So like I mentioned earlier, it's an acute sort of symptom. So in this study, we wanted to look at the acute and short-term outcomes following an injury. And also we wanted to validate a model, chimera, which is short for closed head impact model of engineered rotational acceleration. The key attribute of this long name is rotational acceleration. A lot of popular models for TBI use either fluid percussion or controlled cortical impact or contusion injuries, which require either the skull to be fixed or open, which actually turned out not to resemble what happens in a traumatic brain injury, where cells and white matter tracts are stretched and compromised, and the injury is chiefly achieved by diffuse axonal injury. So the chimera model here that we validated in this study actually does induce diffuse axonal injury. It is a translationally relevant model, allowing the injury to occur in a biochemically controlled as well as reproducible manner. And also, like I said, it's non-invasive, requiring no surgeries. So our experimental design here was that we induced the injury in mice at a force of about 0.6 joules, which seemed to induce a mild, not severe or extreme form of TBI. And the mice that we used here were 7 to 8 months. So if I can demonstrate here, when pressure builds up in the air pressure sort of part of the device here, and you can see when the skull is hit, it is allowed to rotate, so the head is free to move, which accounts for the diffuse axonal injury. So one variable that we're looking at was neuroplasticity, and in particular LTP, or long-term potentiation, as a measure of neuronal plasticity following TBI. Our working hypothesis was that we thought that mild repetitive traumatic brain injury induced by the chimera model would promote neuroinflammation in white matter tracts, confirming the nature of the injury, and would alter plasticity and long-term potentiation. So we focus at the CA1 region in the hippocampal region CA1 here. So a previous study done by Norris and Sheff actually demonstrated deficits in connectivity in the CA1 region. However, it was done at 2, 7, and 14 days marks following the injury. So like I mentioned, we were more focused on short-term and acute outcomes of the injury, and also using the chimera model. The previous study used a contusion injury model. So in order to confirm the diffuse axonal injury, we looked at white matter gliosis, in particular microgliosis using the IBA1 cell marker and astrogliosis using the GFAP cell marker. And as we can see here, this is a representative image of how sort of the quantification was done using ImageJ. And this here is the optic tract, also one representative white matter region that we looked at in addition to the corpus callosum. Also, field excitatory postsynaptic potential were measured in the CA1 region using a 4x4 multi-electrode array probe, as you can see here. And that was done in order to evaluate baseline amplitude, measure synaptic strength, as well as assess long-term potentiation following the injury. So like I mentioned, 2 injuries were induced 24-hour apart. One subcohort that was used for electrophysiology 2 to 3 hours post second injury, very short term, very sort of acute. And then 7 days post second injury, immunohistochemistry and lab staining was performed to confirm white matter injury. So to sum up the results here, we can see that there was increased expression of GFAP and IBA1 in the injured group compared to the group that was not injured, the sham group. And this was demonstrated in the optic tract and the corpus callosum, as opposed to only the optic tract in the case of IBA1. But this was evidence showing that the model, the chimera model, using the parameters that we optimize, actually does induce diffuse axonal injury, leading to white matter inflammation. Looking at neuroplasticity, we were able to see sort of significant differences between the injured group, TBI group, and sort of reduced excitatory postsynaptic potential amplitudes following the injury when compared to the sham or the control group. Another parameter that we were trying to look at was long-term potentiation. So however, like I mentioned earlier, this was done as a summer project. I was just going straight right after sophomore year. So time was a limitation here. The cohort for performing LTP investigations was somewhat small. However, while the data is not conclusive, there seems to be some disruptions in LTP following the injury, which is an indication of overall reduced or altered or compromised white matter connectivity following chimera-induced TBI. So some take-home messages is that our study shows that repetitive mild TBI induced by chimera causes white matter gliosis in the optic tract and corpus callosum, confirming the nature of the injury. Slice electrophysiology also showed that the injury induced by chimera alters LTP levels and significantly reduces excitatory postsynaptic potentials in the CA1 hippocampal region. So further validation of the acute and short-term effects of the chimera model may pave the way for more therapeutic interventions that can alleviate the more lasting and the persistent TBI outcomes, including plasticity deficits, cognitive deficits, and cephalic pain and post-traumatic headache. So I'd like to thank the summer program that I was part of, allowing me to do this work. And with that, I would like to end. If you'd like a comprehensive list of references, feel free to scan the QR code, and I will be happy to take any questions. So thank you so much.
Great, thank you. There actually is a question in there if you want to look in the chat. Since the C57BL mice you used are aged around 7 to 8 months, older than the usual used adult mice, do you think neuroplasticity would be higher in younger mice?
Yeah, that's a great question. Yeah, I would think that plasticity would be higher in younger mice compared to maybe 2-year-old mice. But one reason we could not, like I mentioned, 2 groups that are susceptible are the younger mice and the older mice. We could not incorporate older mice in this experiment using the model because whenever they will be used, we cannot confirm that the injury was actually mild. It can be more severe. So we were not sure of the parameters that we're working at. But I think that's a great question. Younger mice, I think, would have more pronounced sort of plasticity, neuroplasticity. So thank you.
Okay, great. Thanks a lot. So we're moving to the third category now, which is pain. And so first up is Ludovica Brusaferri from the Martinos Center for Imaging at MGH and Harvard. Please go ahead.
Thank you. Can you all hear me?
Yes, we can hear you. You just need to share your screen, please.
Yeah. Perfect.
Okay. Thank you.
Of course. Okay. Can everyone see?
Yeah, perfect.
Perfect. So, hi everyone, I'm Ludovica. Thank you for the introduction. I'm a postdoc affiliated with Harvard Medical School, and I'm going to talk about the impacts of COVID-19 pandemic-related disruptions on neuroinflammation in chronic pain patients. So the research work that I'm presenting is centered around neuroinflammation, which is an inflammatory response within the brain or spinal cord, where glial cells play a key role, undergoing a series of cellular responses that is collectively known as glial activation. So in normal condition, neuroinflammation is a protective mechanism. However, if the resolution phase of the inflammatory response is altered, uncontrolled inflammation might become chronic. And these can lead to many pathological mechanisms, including neurodegenerative disease, such as Parkinson's and Alzheimer's, but also chronic pain conditions, such as migraine. Some of those are still not well understood, but we can use neuroimaging tools to try to understand them better. Specifically, as I'm showing in this slide, we can use PBR28 PET, which is an imaging modality that targets TSPO, a mitochondrial protein whose expression is relatively low under physiological conditions, but it is upregulated during neuroinflammatory states. You can imagine that the higher the inflammation in certain areas, the higher will be the PET signal. But why are we interested in neuroinflammation in the context of the COVID-19 pandemic? Well, since the pandemic onset, an increased prevalence of fatigue, dyscognition, such as brain fog, and other symptoms has been reported, including among the non-infected. And a wealth of preclinical studies have shown that inflammatory process and social behaviors are deeply connected. The inflammatory process can affect social behaviors, and also exposure to chronic stressors can upregulate inflammation. Therefore, both clinical and preclinical literature have raised the possibility that neuroinflammation might be a potential mechanism underlying those symptoms. In our pilot study, we have in fact demonstrated that otherwise healthy subjects who were not infected with COVID-19, yet underwent psychological distress, as many of us during the pandemic, exhibited higher levels of multiple neuroinflammatory markers with respect to those examined before the pandemic. Here, I have reported the mean PET images for both groups, and you can clearly see an overall signal increase in the pandemic group. So now we extended our investigations to chronic pain patients, specifically chronic low back pain and migraine, both conditions previously linked to neuroinflammation in the pre-COVID era. The rationale for this was to better understand the impacts of pandemic-related disruptions on pre-existing neuroinflammatory conditions. So 97 PET MRI images of subjects from both pain populations were analyzed retrospectively. And pre-pandemic and pandemic PET images were compared against using non-parametric permutation inference, which produces a statistical map of voxels that are statistically higher in one group with respect to another. Participants from the pandemic cohort showed increased PET uptake in the pandemic groups from both pain populations, overlapping within a cluster that is encompassing the anterior cingulate cortex, amygdala, insular cortex, and frontal orbital cortex that I'm showing in the last row. Some of those areas have been previously linked to either stress or pain. Moreover, if we look at the average trend over time in panel C here, we can notice a slight descending trend in the last month, which is hopefully suggesting that the signal is returning to baseline. Of note, we also observed a negative correlation between TSPO metrics and pain clinical variables, specifically migraine frequency for the migraine group, suggesting that those TSPO elevations might reflect anti-inflammatory or protective mechanisms. Overall, this study has implications for better understanding the mutual interactions between social stressors and pain, and possible exacerbation of pre-existing neuroinflammatory disease, including migraine, and possible changes in the baseline markers of ongoing or future clinical trials during the COVID-19 pandemic. Thank you.
Okay, great. Thank you very much. So our next speaker in the pain category is Giuseppe Cataldo from Queens College, City University of New York. Please go ahead.
Can everybody see my screen?
Yeah, we can see the slides. You might want to put them into slideshow mode. Yeah, perfect.
That worked?
You had it there for a second. You just click it one more time. Yeah. Yeah.
I don't know why it keeps going back.
Yeah. Okay. It's okay. We can still see the slides.
Thank you. My name is Giuseppe. I'm a postdoc at Queens College in New York in Joshua Bromberg's lab. So I'm going to talk about this PRXL1 knockout mouse as a model for chronic pain. And since I was invited to this, its implications on migraine. So a little introduction, a little background first. We've talked about trigeminal neuropathic pain. It's this debilitating orofacial pain. It's more prevalent in women, also with age. It's difficult to manage. It's resistant to conventional treatments, and as has been said, this kind of chronic pain contributes significantly to a poor quality of life. It doesn't kill you, but it really wrecks your life. So this knockout animal that we have, this PRXL1, it's a homeodomain transcription factor. It's indispensable for the development of patterning the trigeminal lemniscal pathway. This is typically the pathway of fine sensory information transmitted from the periphery to the brain. It's also expressed by sensory neurons in the dorsal ganglia in the dorsal horn. This is somatically, but PRXL1 deletion, we found it disrupts the patterning of primary sensory afferent fibers into the spinal dorsal horn, but it also abolishes the patterning of the whisker to barrel cortex, this pathway, which I'll show you in a second. So since it's been about 20 years where it's been known that PRXL1, this deletion, is associated with hypoalgesia, which is less pain to the body. We've recently found that this deletion trigeminally, facially, is associated with hyperalgesia. So this is what the patterning looks like. So here we have the PRXL1 knockout animal. As a control, we have what's called a barrelless animal that has a spontaneous mutation where you lose the patterning just in the cortex. And obviously in the wild type, the patterning is intact. So in the PRXL1 knockout, you lose this patterning, as you see on the right, in the PRV, the primary sensory nucleus. It's somewhat intact in the SPV, or the spinal trigeminal nucleus. And you lose it in the thalamus and the cortex. So this patterning produces like a perturbation of the trigeminal pathway, basically from the periphery, from the whiskers to the brain. And what does that do? So some of the methods we used, we looked at tail flick, which is thermal pain, and we also used a von Frey, which is a calibrated nylon filament. You apply it to the hindpaws of the animal 10 times, the animal withdraws. The higher the frequency of withdrawal indicates an increase in pain. And reverse the latency of the tail flick, the longer it takes the animal to move its tail from a radiant thermal heat, that also indicates a decrease in pain. So here we have the somatic pain, which was known. This is the body pain, the tail and the hindpaws. You see that the PRXL animals show this hypoalgesia, this less sensitivity on the body. And so this is the news, though. In the trigeminal pathway, facially, we found it unexpectedly, we found the opposite. Now here what we do is, I show the face of the animal, the whisker pad. We apply the same von Frey to the whiskers, to the vestigial pad, and you see here that the facial withdrawal frequency of the PRXL1 animals is significantly greater than VRL or the wild type. And so that's the behavior. We also did some electrophysiology. And so we're doing extracellular in vivo recordings from identified what's called a wide dynamic range neuron. This is a nociceptive neuron in the principal sensory nucleus, which I'd said earlier. The PRV is typically, it's the pathway that's been known for fine touch, not the nociceptive pathways, which I'll show you in a second. The nociceptive pathway is this SPV, the spinal nucleus, has typically been associated with pain. And you see here, when I say identified wide dynamic range neurons, these neurons, when you stimulate them, they respond to brushing, they respond to pressure in a graded fashion, and they also respond to pinch. Whereas what was believed to be in the lemniscal pathway is the fine touch, which only responds to a brushing, but does not respond to pressure or pinch. And then further, we found that the neurons that are in the PRV, which are not supposed to be pain neurons, show sensitivity by an increase in spontaneous activity, which is typically seen in sensitized neurons that show hyperalgesia. And on the right here, we show this new idea of the lemniscal pathway, this low threshold pathway, this low threshold touch with the WDR neurons that we routinely find now, as well as the paralemniscal pathway. This is typically the noxious input pathway. So in conclusion, we found that this, I missed the carprofen. When we did the trigeminal pain, we also gave these animals that were hyperalgesic, they were in pain, we gave a simple NSAID to these animals, and we found that it worked. It actually attenuated the pain associated with this disruption of patterning. So in conclusion, this attenuation of this oral hyperalgesia that we found is consistent with animals that are in a pain state. So what this suggests is that this disruption of this lemniscal patterning in this PRXL1 knockout, it's selectively associated with orofacial pain, and these altered behaviors are reflective of a chronic pain condition. And ultimately, this mutant may be a useful non-invasive model of orofacial pain. But what does this have to do with migraine disease? What are the implications of this research for understanding migraine disease? And since the early work in the 40s by Harold Wolff, it's been known that intracranial structures such as the meninges with the blood vessels, including more recently the dura, more recently the pia, are pain sensitive, and they can show referred pain extracranially, basically on the skin and the skull. This could account for headache and migraine. More recently than that, neuroanatomical, neurochemical studies have shown that most sensory fibers from intracranial tissue structures and extracranial tissues originate in the trigeminal ganglion. So our PRXL1 knockout with its facial hyperalgesia by a perturbation of the trigeminal nerve and the trigeminal ganglion, we've shown this as a new model of trigeminal neuropathic pain. But given that this form of pain, trigeminal neuropathic pain, has been previously associated with migraine, the implication of this is that it may be a novel mouse model for migraine disease, leading to maybe new avenues of study. And so with that, I'll take if there's any quick questions. Thank you.
Okay, great. Thanks a lot. Doesn't look like we have any questions, so I think we could head to our next speaker in this category. But if you've had any questions, we can still send them in, and we can get to them after the next speaker. So the last speaker in our pain category is, why don't we then move on to the first talk in the next category, which is special sensory / comorbidity. And first up will be Hannah Martin, who is at the University of Chicago, I believe. Hannah, please go ahead.
Hi, I'm Hannah Martin. I'm a fifth-year PhD student in Ruth Ann Etock's lab at the University of Chicago. And so I'm a vestibular researcher. I study the vestibular sense, which is integrated downstream with many other sensory modalities like vision and proprioception to provide gaze and posture stabilizing reflexes. And in mammals, this vestibular input comes from the inner ear, and there are 5 end organs in the inner ear. We specifically study the utricle, and the utricle is full of thousands of receptor hair cells that detect head motions and gravity and send this information all over the brain. So it goes cerebellum, brainstem, cortex, where it is integrated with these other modalities. And what we study is really the hair cell level. What is the information that is coming in to these hair cells, and how are they encoding this information before sending it downstream? Specifically, we are interested in the differences between type 1 and 2 vestibular hair cells. So type 1 hair cells are enveloped by this large calyx synapse, while type 2 hair cells have really tiny bouton-like synapses. They also really differ in their voltage-gated potassium channels. Type 1 hair cells have huge low voltage activated potassium channels, whereas type 2 hair cells have smaller inactivating A-type channels. our lab focuses on these potassium channels because they really are dominant forces in shaping the size and time course of the receptor potentials that ultimately drive synaptic transmission, and because they are huge sources of potassium flow, and ion homeostasis is a huge area of active research in disorders of the inner ear. So that's more Meniere's disease, which can be mistaken for vestibular migraine. So we first sought to identify the genes and subunits underlying these two hallmark potassium channels, and following a long history of investigation, our primary candidate was voltage-gated subunit Kv1.8. So we characterized the potassium currents and vestibular function in a mouse lacking Kv1.8. We used immunocytochemistry to show that Kv1.8 is at the basolateral side of these vestibular hair cells, and we used electrophysiology to show that type 1 hair cells and type 2 hair cells both lack their distinctive conductances when you lack Kv1.8. So this shows that Kv1.8 is a necessary subunit of GKL and GA. Moving forward, we characterized receptor potentials, and we found that Kv1.8 is important for reducing the size of receptor potentials. So these are smaller receptor potentials, and making those receptor potentials very fast. So making them occur quickly. And these are expected results based on Kv1.8's impact on resistance of the membrane. And it has important consequences for how quickly receptor potentials can encode head motions. Based on this, we predicted that Kv1.8 null mice would have specific deficits in their vestibulomotor behaviors, or ability to control their body while they're moving through space. And indeed, Kv1.8 null mice, partially because of these deficits in high-frequency stimulus encoding, had abnormal swim posture, they had trouble crossing a narrow balance beam, and they spent less time on their hind legs, less time in an unstable position. Altogether, these results have related a hair cell mutation in an ion channel to the receptor potential and their vestibular motor behaviors. So these findings are very much in the periphery. This is about the sensory encoding of stimuli. And our findings can benefit specifically the vestibular migraine field in two major ways. In the first, there are many unknown mechanisms at play in early vestibular processing. If we hope to apply drugs to treat ailments, we need to understand how they actually impact the circuit. To that end, we've elucidated molecular players in the vestibular inner ear, and this will help us uncover therapeutic targets that may allow us to dial up or down vestibular input to the brain. In a second way, and something that is pretty exciting for me lately, is that vestibular system symptoms are difficult to assess both clinically and in mice. So developing better tools to assess vestibular function is a growing interest. So we've just begun applying some machine learning tools to analyze this rich video data set, where we can look at, we have this mouse with this mutation, and then we can analyze what is its vestibulomotor pattern like. So I'm really excited for that and see where that tool development takes us. But yeah, I've really enjoyed everyone's talks so far. Thank you.
All right, fantastic. Thank you. So it looks like we have Ivana back. So Ivana, if you'd like to try and share your screen, and we'll see if it works now.
So I am a first-year master's student at Trent University. This is my first time at an event like this, so I just pulled together some information. Hopefully that's enough. I apologize if it's a bit yeah, okay. So this is on my poster from SFN, and we looked at how amygdala kindling alters pain sensitivity and pain-related emotional behaviors in rats. So our research aimed to investigate the experience of pain in individuals with epilepsy. And we are particularly interested in this because of high comorbid pain conditions, such as migraines. So to investigate this phenomena, we used a rat animal model referred as amygdala kindling that reproduces the changes that occur in the brain after repeated experiences of seizures. And it does this by passing a current through the basolateral amygdala, the location here. We would implant an electrode that would pass a current at this point. And with each stimulus, it progressively lowers the threshold for excitability. This change in threshold begins in the amygdala, but then it gradually spreads until it reaches the cortex, and the rat can be considered kindled. So in our study, we were particularly interested in the behavioral response to pain and the development of avoidance of pain. And we hypothesized that we could find a higher pain sensitivity in seizure rats than in non-seizure rats, indicated by a greater avoidance of pain and a larger behavioral response to pain. So to study this, we used an aversive stimuli, specifically inflammatory pain. We injected formalin into the hind paw, and we paired that formalin with one of two distinct chambers. So we can see here on the slide, the left chamber, it has distinct features. The floor of the chamber on each side are different, the color, and we also use different scents. We apply different scents to each chamber. So one chamber was paired with the aversive stimuli, and it was referred to as the paired chamber. The other chamber had the absence of the aversive stimuli and was referred to as the non-paired chamber. And aversion was determined by the difference between the time spent in the paired and the non-paired chamber when the animal was allowed to roam freely between the two. So we found that our seizure rats did not develop a significant avoidance of the formalin, while the controls did. But further examination revealed something more interesting. So here we can see on the left is just a visual of the CFOS cell staining. Kindle just refers to seizure rats. Sham are rats that have not experienced seizures. You can see that, interestingly, there is a decreased activation of the medial prefrontal cortex in male kindled rats specifically when compared to the other rat groups. While the other rat groups didn't seem to have a significant difference between either of them. This is also a representation of that divided by their subregions. Sorry. Seizure rats also showed a decrease in overall pain behavior. We were also able to confirm a higher activity in the anterior cingulate cortex in seizure rats, indicating that the seizure rats experience pain more intensely than the non-seizure rats. We were able to negate this then by inhibiting the ACC, which is seen here in these two columns. And it produced an analgesia accompanied by the development of a preference. So you can see here that the rats actually spent more time in the paired chamber, which is paired with the aversive stimulus, than in the non-paired chamber. And another interesting finding that we found was that female rats, they actually produced a higher avoidance than the male rats did overall. So generally, we were able to find that seizure rats experience pain more intensely than non-seizure rats, and that seizure rats have more difficulty in managing their response to the painful stimulus. The results of this project opens various avenues for investigation in the experience and the management of pain in those whose brains are susceptible to overactivity. The difference in experience of pain between the male and female rats also provides interesting potential for further research. The findings of this research can also relate to migraines potentially because it provides insight into how pain is managed and avoidance is developed in those whose cortical excitability is higher than normal, a state that can describe both those who experience migraines and those who experience seizures. Our research may also provide an avenue through which comorbid anxiety and panic disorders can be better understood due to a characteristic of amygdala kindling model producing fear and anxiety related behaviors. Thank you.
All right, great. Thank you very much. That was fantastic. So I believe we have 2 talks left. We're back to our final category, special sensory / comorbidity. And next will be Asan Mary Cedras, who is from the University of Montreal. Please go ahead.
Hello, everyone. I'll just try to share my screen. Do you see it?
Yes, perfect.
Okay, perfect. So hello again. My name is Hassan. I'm from the University of Montreal in Canada. I'm a first-year PhD student in Dr. Meyer's lab. And today I will talk to you about a study that we did in our lab, which is titled Investigating the Relation Between Vestibular Reflex and Self-Motion Perception May Help Distinguish Meniere's Disease from Vestibular Migraine. This is a preliminary report. So differentiating between Meniere's disease and vestibular migraine, which are 2 vestibular pathologies, is a major clinical challenge. Now, why is that? First of all, the diagnosis is only based on the symptoms reported by the patient. However, the symptoms in vestibular migraine and Meniere's disease significantly overlap, as both can experience hearing loss, tinnitus, dizziness, and migraine. Finally, there is no objective measure that is specific or sensible enough to dissociate between these 2 pathologies. Now, the objective of my study was to compare the vestibulo-ocular reflex and perceptual response between healthy participants, participants with Meniere's disease, and participants with vestibular migraine following a vestibular caloric stimulation. A caloric stimulation is a test used in clinical practice to evaluate the vestibular system by putting water into the ears. This test is also known to induce a rotation sensation for about 1 minute. Here is a picture of how we placed our participants. They were lying on at a 30-degree angle. We used hot water for the stimulation. They also had goggles to record the eyes movement velocity, which is the response of the vestibulo-ocular reflex. And they also had a hand wheel for the perception data. All the participants of the 3 groups that is listed in this table as to perform a task where we ask them to turn the hand wheel as fast as they felt they were rotating following the caloric stimulation. For our analysis, we looked at the eyes movement velocity recorded by the goggles, and we also look at the perception of induced vertigo with the data of the hand wheel. Now, for our findings and conclusion, first of all, I want to say that we didn't find any significant differences when we tried to correlate the vestibulo-ocular reflex with the perceptual data. But where we find significant differences is here in this picture, and I'm gonna explain it to you. When we look at the vestibulo-ocular reflex alone at the right part of the graphic, we can see that people with Meniere's disease, they have a significantly slower eyes movement compared to controls. Now, where it is getting more interesting is when we look at here at the right side, when we look at the perception data, we can see that people with vestibular migraine felt they were rotating faster compared to controls, but more interestingly, compared to people with Meniere's disease. These results, in conclusion, demonstrate for the first time that motion perception following caloric stimulation may help dissociate Meniere's disease from vestibular migraine. Now, what does my study bring to the migraine field? First of all, the results of my study are inviting future studies, to pay more attention to motion perception, more precisely to self-motion velocity perception when studying vestibular migraine, as it may be a factor specific to this population. My study provides also an objective method that could help us differentiate vestibular migraine from other pathologies with similar symptoms, such as Meniere's disease. And this objective method is an easily applicable one to assess self-motion velocity perception in the vestibular migraine population. Like I said before, caloric stimulation is a tool that is often used in clinical practice either by audiologists or ENT doctor. So thank you all for your time.
Great, thanks very much. So I believe we're now at our final talk. Next up is Shafaqat Rahman, who I believe is at the University of Rochester. So please go ahead.
Hello, can you hear me?
Yes.
Cool. Hi, I'm Shafaqat. I'm a PhD student in Anne Lupke's lab at the University of Rochester, and I'm here to talk about calcitonin gene-related peptide and motion sensitivity, and thus its implications for a disorder like vestibular migraine. Here's a picture of our beautiful campus when we're not stuck in 10 feet snow, 75% of the time. So why CGRP? CGRP causes attacks in people with a history of migraine, but not healthy controls. And since its preliminary research, the FDA has approved many therapeutics that block CGRP. These therapeutics are very effective based on recent clinical trial data. But a common misconception of migraine is that it's just a recurrent headache. Migraine is more than that. It's a disorder characterized by hypersensitivity to various stimuli. Patients with a history of migraine but experience sensitivity to motion may have vestibular migraine, which is the #1 cause of vertigo in dizziness clinics and affects about 3% of the US population and affects 3 times more women than men. So my job as a preclinical scientist is to look at preclinical surrogates to research that emulate clinical symptoms. And what our lab looks at is imbalance and motion-induced nausea through the center of pressure test and the thermoregulation to provocative motion assay. Okay, why is it slow? So the center of pressure test is used as a surrogate for imbalance. And the rationale is that in the clinic, we ask patients to stand on a force platform, and the forces that their body directs into the platform create a center of pressure ellipse. When a person is in control of their balance, you can imagine that the ellipse is tight and small. Now ask the patient to do a challenge. Have their eyes closed or stand on 1 foot versus 2. What you'll see is the ellipse gets bigger and suggests an imbalance. We study mice similarly and are interested in observing what happens if we give them CGRP or challenge them with a vestibular stimulus. So let's look at the methods. The methods involve treating mice with saline or CGRP 20 minutes prior to testing. We then take 10 measurements of the mouse with all feet on the platform. And after those 10 measurements, we rotate them with a vestibular challenge. After that rotation, we take another 10 measurements. And here's some representative pictures of what we should expect the ellipse to look like during the control measurement. If we look at the actual data, in males, when you challenge them with CGRP or give them a vestibular rotation, they more or less don't respond to either challenge. But then when you look at the females, if you inject them, or if you give them the vestibular challenge, a few of the females will respond with higher ellipses compared to their pre-vestibular challenge state. And each ellipse that we're seeing here is not just one measurement, it's the average of 10 measurements for that condition. Now, those same females, if you inject CGRP, what do we see? We see that the center of pressure ellipse, the average center of pressure ellipse, increases significantly before the challenge and maintains that within that range after the challenge. And it shows that this observation shows that IP-CGRP may have a sex-specific effect in females, and that's related to imbalance and postural control. Now let's look at another assay, thermoregulation as a surrogate for motion-induced nausea. Now the rationale for this assay is unique because mice cannot vomit. They don't have an emesis reflex and they can't vocalize to you that they're nauseous. So we have to look at interesting, unique ways to explore this symptom. What we do know is that between rodents and humans, we share a hypothermic response to provocative motion to nauseating stimulus. And that in rodents, because they have a tail spike, they actually respond to a nauseating stimuli with a transient tail spike. So if we look at the methods, again, we inject mice with vehicle, CGRP, or with blockers. We get a 5-minute baseline of their baseline temperature, no challenge whatsoever. We then rotate them for 20 minutes, measure their temperature fluctuations over time, and then measure their temperature again at the level of the head and the tail during the recovery period after the rotation. We've done the test to retest reliability first, and we see that within a given mouse, you can actually recapitulate this phenomena 4 to 6 days in between tests. And so it's a cool assay to explore. Now let's explore this with CGRP and with blockers. Females are in orange and males are in blue. In the vehicle control state, we see this healthy response to provocative motion. Both either sex, males and females respond. They show that tail spike that goes up and down within 10 minutes. Treat those mice with CGRP, and what we see is we don't see that response to the rotation. The mice might be grimacing, but they're not responding to the rotation with that tail spike. Now, if you take a subset of these mice, give them either olsegapont, sumatriptan, or rizatriptan. Olsegapont, as many of you know, is a CGRP receptor antagonist, and it blocks CGRP signaling at the receptor level. And sumatriptan and rizatriptan are selective serotonin receptor agonists that may modulate CGRP indirectly. These tryptin drugs are commonly used in the dizziness clinic to treat migraine, but are known to not really affect vertigo. So what does that mean for our results? If you see in the males, when they're given olsegapont delivered with CGRP, they actually respond to the provocative rotation, and the females have a harder time. Both triptans that we know don't work in the dizziness clinic are also not working in our preclinical model. If you look at the tail vasodilations across all animals, again, more males respond when they're co-administered with olsegapont, and a handful of females also respond with olsegapont, but we don't really see that effect with the triptans. If you look at it a percentage, 80% of male mice respond with the receptor antagonist to CGRP, and 43% of females show that rescue, but lower efficacy is seen with the triptans. So this data mirrors clinical findings in the dizziness clinic. These are not effective for vertigo, motion-induced dizziness, and maybe a CGRP antagonist may have a better role for this therapeutic strategy, as a therapeutic. So my concluding remark, in summary, our findings suggest CGRP sensitivity plays a role in DM symptoms of imbalance and nausea. And future work will explore the role of this peptide in novel mouse models with modified receptor signaling. And if you're interested in asking me more about what models we're looking at, do send me an email. But since we're talking about related comorbidities, me and my advisor, Dr. Lupi, we've also been involved in a project studying CGRP's role in COVID-19. And patients with COVID-19 can have vestibular issues. A recent paper actually in September suggested CGRP in sustaining COVID-19 related hyperimmunity. So we studied COVID-19 in mouse models, and we recapitulated severe COVID symptoms in 2 mouse strains, the C57 wild type and the 129 wild type. We observed infection to cause fever and weight loss, increased stimulation of IL-6, lung inflammation, and infect motion and dizziness. And we saw that the receptor antagonist olsegapont reduced IL-6 levels compared to placebo-treated mice. And it also improved weight loss recovery in older but not younger mice, without really much improvement on lung inflammation or dizziness outcomes. So it suggests that maybe as CGRP antagonists may have a role in treating COVID-19, and maybe we should repurpose a migraine drug for this pathology. That's all I have for today. Thanks for listening. I'm always happy to discuss further.
Okay, great. Thanks so much for that. So I believe that is all the presentations. If I forgot anyone, please shout out now or forever hold your peace, but I do believe that that is everyone.
So I also should just explain a bit about the Association of Migraine Disorders, because this nonprofit was started by otolaryngologists. And so for our focus was to look at the sort of non-headache forms of migraine disease. And we've been concerned that the general medical community and the public don't understand that this is an inherited brain dysfunction, and that it's much more than a headache disorder. So in our field, it's a major factor causing the misery of dizziness, imbalance, hearing loss, and tinnitus, abnormal olfaction, and other special sensory disorders. The disease also is not just a neurologic dysfunction, but a breakdown in the immune and cardiovascular systems. But the issue that's most dear to me is the preponderance of patients that I see struggling with chronic or recurring rhinosinusitis. And at the moment, the medical community has got this all wrong. Each year, 15% of the population is diagnosed with rhinosinusitis. And this condition is associated with roughly 73 million restricted activity days, with direct medical costs of almost $2.4 billion Antibiotics are prescribed in 82% of office visits for presumed rhinosinusitis, making it one of the top 5 most common reasons for using antibiotics. And yet, patients with clinically suspected rhinosinusitis, only about 1/3 of them have a bacterial etiology. It's becoming increasingly clear that over 60% of these 50 million people suffering from are suffering from a sensory illusion or hallucination created by a chronic dysfunction of the trigeminal and autonomic nervous systems. This indiscriminate use of antibiotics raises concerns about accelerating allergic reactions to antibiotics, complications caused by C. difficile overgrowth, microbiome changes in the gut, unnecessary costs, and bacterial resistance. Every piece of information about how this pathophysiology works in the trigeminal nerve and autonomic system is critical to rewriting the story about rhinosinusitis. So as we bring the evening to a close, it's been my honor to host this group of researchers. And when they wonder why their work might have any use to the community of migraine patients, I remind them that any contribution that leads to improving understanding or managing migraine is going to have an impact it's going to impact a lot of people, because migraine disease affects more people than all of the other major neurologic diseases combined. That is at least 30 million Americans. And if you think we have this disease licked when you watch the latest splashy commercial from Khloe Kardashian or Venus Williams, remember that even with the new CGRP blocking medications, more than 50% of those with migraine feel that their treatment is ineffective. There remains a golden opportunity to discover a biomarker to improve diagnosis or other key therapeutic target, particularly with a high prevalence of vestibular migraine, brain fog, sleep, and mood disorders, which are likely mediated by dysfunctions in the brainstem, hypothalamus, amygdala, cerebellum, and parabrachial nucleus, and the inability of monoclonal antibodies to cross the blood-brain barrier. New targets and formulations need to be discovered. And we can only do this with more communication and collaboration between research labs and a broader understanding of how a new revelation about a hormone receptor, an ion channel, a piezoreceptor, or a transcription factor can be that translational key to a new migraine therapy. So in closing, I will hand it back to Neil Andrews, who can explain his role as editor of the Migraine Science Collaborative. But again, thank you for all the participants.
Great, thanks, Rick. Can everybody hear me? Everyone hear me? Okay, great, yeah. So as the judges are doing the work now, we have a bit of airtime to fill. So Rick had just asked me to say a few words about the Migraine Science Collaborative. So I run the Migraine Science Collaborative. I'm the executive editor, and I'm a science journalist by background. And basically, I know you're all familiar with MSC, but hopefully I can tell you a few things you might not know. But we relaunched it with a brand new website that you can see here. We relaunched it actually very recently, just a few months ago. And the goal is really to provide a central place or hub where the international migraine community and people who are doing work in ancillary areas can come to gather for the latest news, information, events, resources, and so forth. And the goal is really to build that community, and including with events like tonight, as Rick was saying, to really bring people in from adjacent fields who really may not be working on migraine per se, but are doing a lot of work that has tremendous relevance to migraine. As you all know, Rick was talking about how large a problem migraine is. As you all know, funding for migraine research is not great. It could be a lot better. And one of the big picture goals, the big things for Migraine Science Collaborative is really to raise the visibility of migraine build that community so that there's more attention in society, general society, to migraine. It raises the profile of migraine, raises the profile of the research that people are doing, and hopefully that attracts more attention to the field and builds the field, and maybe one day that will even increase funding for the field. So that's sort of the lofty goal. In terms of the day-to-day, we are covering the field for researchers and clinicians. So we have a small army of really great professional science writers who write news for us. So if you go to our news section, you can see all these different articles we've written on recent papers. Usually each news article focuses on a single paper. Sometimes we'll cover a couple of papers, but this is sort of one of the bread and butter things that we do. Again, really covering the important research for researchers. We do interviews as well with leaders in the field, as well as podcasts, which sort of just gives, people the opportunity to hear the voices directly from, the leaders in the field. So we spend a lot of our time doing those things. I wanted to draw your attention, if you're not familiar with it, to our bi-weekly literature review. So this is basically a curated list of papers that we publish every 2 weeks. So we basically go through the literature. So we're basically doing a PubMed search for you, so you don't have to do it yourselves, trying to save you some time. And it's a compilation of what we feel are the best and the most important papers that have been published every 2 weeks. And you can basically search. So if you're interested in animal studies, you can click on animal studies and you'll see all the animal studies grouped together. We designate a couple of papers or a few papers every couple of weeks as editors' picks, papers that we think are particularly important to the field. It's a good place to start, I think, especially if you're new to the field. And again, we have some categories. So if you want to see a review, if you want to see original research, you can do that. Another thing is we have some professional resources, of which the virtual poster contest is one. We have an events calendar, we have a grant section. So if you're looking for funding, you can check it out. A researcher actually told me the other day, they went to the grant section and they didn't realize that a particular organization actually provided grants for migraine research. So that was great to hear. So please check it out. If you know of any events or grants that you want us to highlight on the site, please just get in touch with me and let me know about that, because we just sort of want to build this. And just also wanted to mention, we're doing some multimedia stuff. So I mentioned some podcasts. We actually have a great podcast with Greg that I had the honor of doing with Greg Dusser, who's attending. So I just wanted to give a shout out to Greg and give a shout out to that. We're also working on doing data visualization. So we've done a couple of these and we look forward to developing sort of new features in the future. If you're not already members, please join. So AMD is a nonprofit. So MSC is a nonprofit as well. And so everything is freely available, but we still love it when people register, when they register to become a member. Becoming a member is free. What it does is it gives you the ability to contribute content and to comment on content. So if you listen to a podcast or read a news article on the site and you want to, provide an opinion, a commentary on that, you need to be a member to do that. But I think even more importantly, the more members that a site like Migraine Science Collaborative has, the more visible it's going to be. Again, it really helps to build the community. And if people can go, oh yeah, look at Migraine Science Collaborative, look how many active members are interested in migraine and doing migraine research, that is a very important thing. So we hope that you will become a member, and so that you will appear in our member directory if you're not already a member. So anyway, so that's just sort of a few quick things I wanted to say about MSC. You can also sign up for our newsletter as well. So this is just a monthly digest of our newly published material each month. We don't spam you, it just comes once a month, and it's just sort of a digest of everything that was newly published over the last month. So you can sign up for the newsletter when you actually register for the site as well. So that's just a quick run through of the site. And I'm just curious if anyone has any questions about the site that you might like to ask me, types of content you might like to see, anything along those lines.
Okay, so I know that Alicia is probably trying to figure out how to get back in here. to announce the winners, but we
I am
do.
actually right here and I am
There
ready
you
to
are.
announce
You're
the
on.
winners.
All right, great.
Yes. So thank you everyone for your presentations. They were terrific and it was really, tough trying to pick some winners here, but we appreciate everyone's time. We know it's late too, so I'm going to get right to it. So for the topic of migraine, the winner is Micah Johnson. Congratulations. And I'm going to go on to the next. Bunch of scrolling. Please bear with me for a moment. For physiology, the winner is, and I apologize ahead of time because I know I'm probably going to butcher your name, but Seifaldin, Selim, Aldalil is the winner in physiology. Congratulations. In pain, the winner is Giuseppe Cataldo. And for special sensory, the winner is Shafaqat Rahman. So congratulations, everyone. And with that, I will hand it over to Rick for some final comments.
Sure. So thank you again for making this all happen. I would mention that reinforce what Neil was saying about the Migraine Science Collaborative. We would like to think that this could be a way of connecting the community in the future. So it's only going to work if people find that it's useful and interesting to them. So if there's anything you can think of that you'd like to see on that site or any contributions you want to make, please reach out to us. We hope that we'll hear from either you or your labs again next year for the third annual contest. And have a good night.